Hello, everyone. Welcome to our webinar. I'm Maya Lim, and I'll be a moderator and co-host for this webinar session. My role at RoosterBio is to serve as a bioprocess consultant for our customers in the West Coast and APAC regions. And I have over a decade of experience in stem cell bioprocessing, tissue engineering, and bioprinting. Now, joining me today is my colleague, Tim Olsen.
Hello and welcome, everyone. I'm Tim, and I will be guiding you through today's webinar presentation alongside Maya. I am a former scientist at RoosterBio, turned commercial team lead for the Northeast USA and Europe.
Before we kick off on today's topic, I wanted to go over a few housekeeping items. So first, today's webinar will be recorded and available on demand after the live session. All registered users will receive an email from RoosterBio after the webinar. If you have any questions for us at any point during this presentation, feel free to send them through the Q&A tab at the bottom of your screen. We'll be answering the questions at the end of the session, and if we don't get to answer your question today, we'll be sure to follow up afterward. Now, if you run into any technical issues, feel free to send us a note on the Q&A tab as well, and our support team will do our best to help you. So without further ado, I'd like to kick things off by giving you a brief update on the recent developments in the MSC cell therapy field for COVID-19. Over the past few months, there's been a constant influx of news from companies announcing their interests in developing MSC therapy for COVID-19. And this is primarily to target a condition called acute respiratory distress syndrome, also known as ARDS. In fact, as of April 15, there are 45 cell therapy clinical trials filed for COVID-19, and 70% of these are primarily using MSCs. Amongst the group developing MSC therapies, here are a selected number of companies that I wanted to highlight. Mesoblast and Arthrosis both have a bone marrow-derived MSC therapy product that are already in the pipeline for graft versus host disease and ARDS respectively. Arthrosis now has a FDA fast-track phase 2-3 trial called Macovia, and Mesoblast has initiated their phase 2-3 trial recruiting 300 patients in the US. Hope Biosciences currently have 4 FDA-approved trials for COVID-19 using their adipose-derived MSCs both for autologous and allogeneic use. Pluristem has extended their compassionate use under CTAP, which is the Coronavirus Treatment Acceleration Program, for their placental-derived MSCs. Olsen Therapeutics has initiated a multi-center clinical trial in the UK for their umbilical cord-derived MSCs. And both Citius and Sonata are working on iPS-derived MSCs, and they're currently in preclinical development stages for COVID-19. Last week, Citius announced they're initiating their pre-IND discussions with FDA to submit a trial under CTAP. Now, also to highlight here are some of the prior and early clinical data reported so far in the use of MSCs for treating ARDS. The group that has by far done the most significant amount of work in ARDS is Dr. Michael Mathey from UCSF, who has completed a phase 2A trial and reported data from 60 patients, demonstrating safety up to 10 million cells per kilogram. Other promising updates have been reported by Pluristem and Mesoblast, showing high survival rates of patients who have been treated with MSCs, and in some patients, they're observing signs of respiratory improvements. Now, clinical studies performed in China have also observed similar positive results so far. In support of this clinical work, numerous preclinical studies, both in vitro and in vivo, have been conducted by various groups. The goal is to understand the possible mechanisms of action of MSCs on acute lung injury, mimicking ARDS. One of the primary observations here have been MSC's anti-inflammatory effects that is believed to be paracrine in nature. Additionally, MSCs have shown to improve alveolar permeability. This is observed by the restoration of endothelial and epithelial barrier integrity and fluid clearance in the lungs. Dr. Maithai's group has also shown that one of the key contributors of this is the secretion of keratinocyte growth factor, KGF. MSCs are also thought to have an antimicrobial properties and are responsible for reducing apoptosis in some endogenous cells in the lungs. So far, preclinical evidence is promising in demonstrating the use of MSCs and their secretomes for treating ARDS. In view of this pandemic, our team has gone through the process of calculating how many cells would be needed to meet the demands to treat patients with COVID-induced ARDS. As of May 1st, there are already over 1 million confirmed positive COVID cases in the US. Based on these statistics, we know that at least 1% of these patients will suffer from ARDS, so that will be around 10,000 patients. If each patient dose is roughly 300 million cells, we would need to produce 6 trillion cells to treat 10,000 patients. Now, current 2D planar culture platforms, primarily using cell stacks, are limited to lot sizes of less than 10 billion cells. In fact, the typical process only generates roughly 2 to 3 billion cells over 30 days. 3 billion cells can only be used to treat 5 to 10 patients, so in order for us to reach 6 trillion, we would need to run 2,000 manufacturing lots. And currently, there's simply no GMP facility today that can meet this huge manufacturing demand. At 2 lots per week, it will take 18 years. And even if you had 10 GMP suites running simultaneously, it may be realistic to get at most 240 manufacturing runs in about 6 months. So completing 2,000 manufacturing lots is a daunting task. It became pretty clear to us very quickly that this is going to be a significant problem for the industry as a whole. So how can we provide an alternative solution, and what might this look like? I'll pass the presentation now to my colleague, Tim Olsen, who will share some of the insights with you of what we can offer and present viable options that RoosterBio can provide to address this urgent and critical need in the short and long term.
Great, thank you, Maya. I will go ahead and take you guys through the next part of the presentation on scaled MSC-based therapeutic manufacturing. Large-scale production of MSCs requires scalable platforms like stirred tank bioreactors. Depicted here are the technology S-curves as it relates to MSC manufacturing technologies. On the y-axis is the lot size of cells to be manufactured, and on the x-axis are the platform technologies used in MSC manufacturing. These are your T-flasks, multi-layer 2D vessels like cell stacks, and suspension bioreactor systems. The curves show where adherent cell production constraints are for each of these platforms. Each technology platform is usually good for a log or two of productivity, after which you move to the next technology platform to achieve the desired lot sizes. T-flasks are great for early development work where a few 100 million cells are required for studies, but are not really suitable for large-scale clinical manufacturing. Manufacturing quickly becomes cumbersome and has poor cost of goods with a large footprint. Multi-layer vessels or cell stacks were an advancement that enabled more cells to be manufactured in the same footprint, boosting efficiencies and cost of goods, but hit a manufacturing plateau at about the 10 to 20 billion lot size. Limited by scale-out capabilities, this is the technology primarily being used in phase 1 and phase 2 MSC trials today. 3D bioreactor technologies represent true scale-up potential, and tremendous advances have been made over the last 10 years with respect to the upstream process and downstream process unit operations to maximize yields, boost productivities, and really streamline processes. Groups have demonstrated runs at the 50-liter scale and beyond, with paths to scale up for late-stage trial and commercial use on the horizon. Here's where cost of goods, economies of scale are achieved. Manufacturing platform evolutions, like these scaled bioreactor technologies, will drive the economics of stem cell supply for years to come, and really enable a response to global health crises like COVID. After discussing with you guys the growing clinical needs and demand for rapid and scalable manufacturing, let's go ahead and walk through this exercise of estimating the target requirements for COVID-19. As discussed earlier, we're going to assume a patient population of 10,000. Based on trial data collected from the literature, an estimate of 300 million cells per dose will be required for treating a lung-related indication from COVID. Straightforward math would dictate the need for 3 trillion cells to treat all of the patients, but the reality is that we need to manufacture 6.4 trillion cells in order to get 10,000 vials of product doses for COVID, with a target dose of 300 million cells reaching the patient. Why is this? Well, you lose product in downstream processing unit operations, quality control, and overfill operations. In manufacturing, it is a general practice to use 50% overfilling to ensure enough product reaches the patient. Adding in loss due to downstream processing, about 30% on average, and QC testing, we arrive at needing an extra 340 million cells per dose or vial, thus reaching 6.4 trillion vialed cells. There are many ways to achieve the 6.4 trillion cells, but not all of them are practical. If using 10-layer stacks, you can only produce 10 billion cells per lot, and 640 lots would be required. A daunting manufacturing problem at the least. No manufacturing facility is out there today that can handle this. The clean rooms that do exist are currently set up for T cells and other autologous cell therapies. Moving into a 50-liter bioreactor system where yields can reach 30 billion MSCs and beyond, you reduce the lot size required to 215 total, which becomes a little more manageable, but not rapid enough for a pandemic response. Manufacturing for a pandemic requires a pandemic scale manufacturing platform, the 500-liter bioreactor and beyond. Existing processes and protocols are in place. There just hasn't been a demand like the present to warrant R&D studies with high costs. At this scale, the number of manufacturing lots is reasonable and attainable, as your lot sizes are 300 billion cells per batch. Urgency is key to development of patient-ready therapeutics, and RoosterBio has been working with customers for the past 6 years to radically streamline the path to translation. If you are working on bringing a COVID therapeutic to the clinic, here is a high-level view of what to expect in terms of project timeline and workflow. Project planning will establish the cell numbers required and the platform technologies to execute your needs, timelines, and begin ordering raw materials. 2 months is typically allotted here. Next, there are 2 parallel paths: execution of getting manufacturing up and running, and regulatory interactions. The tech transfer of RoosterBio's recommended process can take 1 to 6 months, depending on the GMP facility capabilities, of which product manufacturing typically takes 10 days or less per batch, only to be followed up by 2 months of testing and release prior to having doses ready for patients. As it has been seen for COVID thus far, the FDA is working hard to accelerate promising technologies to the clinic at unprecedented rates with fast tracking, compassionate use, and emergency use authorizations. So any of these timelines suggested here for approval, testing, and the clinical approach could certainly be accelerated. RoosterBio is here to work with you to radically streamline your development timelines and translation to the clinic for treating COVID patients. Now that we have discussed a high-level project plan, I want to go over with you some of the tactical moves with RoosterBio that can be employed to accelerate your therapeutic timeline. Manufacturing readiness level is an indicator of the ability of a product to be manufactured, and manufacturing that is ready to implement is critical for a rapid response to pandemic situations. Partnered with consistent starting materials in the cells and media, RoosterBio has GMP-compatible bioprocess recommendations and protocols that are ready to be implemented immediately at your GMP facility for further manufacturing of therapeutic doses. These are processes and recommended lot sizes that RoosterBio has developed and recommends to our existing customer base today. The working cell bank has consistent CMC, or chemistry, manufacturing, and controls, which allows for development efforts to start from the same starting cellular materials in 2D planar systems or 3D bioreactor platforms. Whether the MSCs or extracellular vesicles, EVs, are the product of interest, RoosterBio will work with your group to understand your cell or EV number and dosing number needs, and make process recommendations for your 2D and 3D platforms. It is much faster to implement a 10-layer process, pictured in the top row here, which yields between 6 and 10 billion cells within 10 days' time. Our partner GenCure in Texas has already gone through this tech transfer process and can rapidly move to support your team. We're also working with them to complete the 50-liter bioreactor process depicted below. The 50-liter bioreactor platform yields between 20 to 30 billion cells within 10 days' time. But this technology requires different equipment, raw materials, and expertise that is not present in most manufacturing groups today. However, RoosterBio is here to help with this, to expedite this process with our bioprocess recommendations and protocols. Further scale up to the 500-liter bioreactor is within reach and has not yet been performed simply because there hasn't been a demand like the present situation, and raw material costs exceed $1 million per run. RoosterBio is the only company in the world today with an off-the-shelf and ready-to-deploy inventory of GMP human bone marrow working cell banks and the paired bioprocess media for manufacturing MSC-based therapeutics. Equipped with US FDA master files and an industry-leading characterization profile, RoosterBio's starting materials will streamline your IND submission and clinical translation. The working cell bank vials, pictured on the left, contain 20 million cells per vial and are meant for further manufacturing towards dose formulation, fill, and finish. When using the paired cell and media system, the xeno-free bioprocess media, which is pictured on the right here, is designed to be used in a batch culture process, which means no media exchanges are required. This streamlines manufacturing processes and boosts productivity metrics, which is critical in scaled and economical manufacturing of therapeutics. The bioprocess media is available in both bagged and bottled configurations to meet your bioprocess needs. 4 customers are in clinical trials using RoosterBio's cellular starting materials, with the first patient being dosed in March 2020. And RoosterBio is very excited to celebrate this. Our products are ready to be deployed now, and there's enough inventory available to manufacture 125 lots of 8 to 10 billion cells, plus existing deep master cell bank inventory to replenish any of the working cell banks. I detailed before the manufacturing readiness is critical for a rapid MSC therapeutic manufacturing response for COVID. Here I will highlight a rapid process for MSC-EV production that can be taken into late-stage development and GMP manufacturing immediately. Like with MSC manufacturing, it is much faster to implement a 10-layer process, pictured in the top row here, which yields 30 liters of conditioned media with particle concentrations in the range of about 1 × 10⁻12 particles per ml. Productivity increases in 3D bioreactor systems as higher cell densities can be realized. RoosterBio has successfully implemented the 15-liter bioreactor process, seen in the bottom row here, for programs in clinical trials today, and is working with clients right now to onboard the 50-liter process. Both 2D and 3D processes start with the same starting cellular materials made with consistent CMC in a 4-day seed train, as shown on the left side of the graphic here. For the second passage, the cells are either cultured in 2D or 3D for another 4 to 6 days prior to swapping out the expansion media for EV collection media for 2 to 3 days. I will share more on the EV collection media on the next slide. After the cells are cultured in the collection media for 2 to 3 days, the conditioned media can be harvested and processed according to your preferred downstream processing methods for therapeutic dose formulation. Again, RoosterBio's manufacturing systems scale with your program, and we would be happy to help plan out your clinical program and present our bioprocess recommendations. After highlighting the processes used for efficient EV manufacturing, let's chat about the reagents involved in these processes. The current EV product portfolio is pictured on the left-hand side of the slide. Rooster Collect EV is RoosterBio's xeno-free, protein-free, and chemically defined collection media. EV Boost is an EV reagent designed to shorten and tune the EV manufacturing process. The GMP Rooster vial is the 20 million working cell bank vial used to inoculate the manufacturing process. And Rooster Nourish is the GMP bioprocess media used to expand the cells. And finally, the newly launched Rooster Collect EV-CC is the GMP counterpart of the research-grade EV Collect and expands the only commercially available GMP and MSC cell and media system to include Streamline path to GMP EV production. On the right, you can see the expected yield for 2D and 3D systems, noting that higher particle concentrations are realized in 3D systems because higher cell densities are achieved in a given volume of media. Our systems have attained 1000x increases in EV yields in just a fraction of the time when compared to other systems out on the market today. RoosterBio's EV production solutions are designed to maximize media consumption, to reduce your cost of goods, provide a streamlined process, and ultimately boost productivities as you march on the path to GMP EV clinical trials and commercialization. After being introduced to the products and processes that enable rapid MSC manufacturing, you might be thinking how your group and RoosterBio would work together. As a therapeutic developer for COVID that is implementing MSCs, rest assured that in RoosterBio, you will find MSC bioprocessing experts that not only provide the cells and media, but the high-level bioprocess recommendations that fit your program demand. As you are managing the therapeutic pipeline, keep in mind, RoosterBio's products were designed for the full life cycle of your program. For your early proof of concept and discovery work, to scale up, to clinically and commercially relevant batch sizes. Finally, when it comes to regulatory, at RoosterBio, we coach our clients in explaining that it is important to begin with the end in mind. RoosterBio's GMP products are equipped with FDA Type 2 master files and deep inventory stocks, ensuring your clinical supply chain for the years to come. Together, your company and RoosterBio have the ability to offer innovative solutions and new therapeutic options for COVID patients, and we'd welcome discussions to explore these opportunities further. Are you ready to take your therapeutic to clinical manufacturing for COVID-related indications? Well, RoosterBio is here to support you with our off-the-shelf paired cell and media systems with bioprocess recommendations. Whether you are working in 2D or 3D culture platforms, using MSCs or MSC-EVs as your final product, RoosterBio's systems scale with your manufacturing needs. At this time, RoosterBio has 4 customers in clinical trials using RoosterBio's starting cellular materials, both IND and compassionate use paths. For more information on RoosterBio or to accelerate your path towards a COVID-19 therapeutic, please contact us after the webinar and our team will be happy to discuss further. That concludes the tech portion of the presentation. And now that we have talked about COVID, MSCs, manufacturing, and a plan for a successful COVID response, I want to tell you a little bit about RoosterBio and our home, which we call the Roost. Founded in 2013, our roots are in Frederick, Maryland, where we came to market to create disruptive technologies in the cell manufacturing space. You will see some of our key strengths here at the top of the slide. At RoosterBio, we are all about high-volume MSCs, bioprocess medias, GMP products, scalable EV production, MSC kits, and process design and acceleration. Through our MSC products, manufacturing bioprocess expertise, and our dedicated team, RoosterBio's mission is to fuel the rapid commercialization of scalable regenerative cures. And our global reach is growing by the day, as we support over 200 customers around the world in 26 different countries. Our main areas of interest are in cell therapy, gene editing, tissue engineering, immunotherapy, medtech devices, extracellular vesicles, and the cosmeceutical spaces. Are you ready and hungry for more information on scalable manufacturing processes for MSCs and EVs? Or want more updates on MSCs being used for COVID indications? Or perhaps you're interested in how to plan for lot sizes in clinical and commercial manufacturing. To explore these topics and more, please check out RoosterBio's blog page on roosterbio.com. Our team adds new content very regularly, and we are actively adding pieces on MSCs being used for the COVID pandemic. These pieces are typically a 10 to 15-minute read and perfect for enjoying with your morning cup of coffee. Our most recent blog was published this morning on adding MSCs to the Strategic National Stockpile for emergency use and deployment. And a link to it will be included on the webinar follow-up email. RoosterBio is eager to work with you all to support your development efforts for a COVID therapeutic response. If you are ready to work now or want to talk through a plan of action, please follow up with RoosterBio's team today. Our team of bioprocess experts is happy to schedule phone calls or video conferences to accelerate your program now. The first 5 responders to our call to action will be eligible for a free Rooster kit, which contains the MSCs and media to jumpstart your preclinical evaluation today. If you reference our webinar today, a 10% discount code will be unlocked for your use on a donor screening kit. RoosterBio will send a follow-up email after this webinar. Respond here or simply email us at marketing@roosterbio.com. Thank you all for the time today. We hope it was informative and an enjoyable break from the new normal.
Thank you, Tim. That was a wonderful presentation. So we're going to move on now to our Q&A section. And so the first question that I got here is regarding the 300 million cells per patient. I think, Tim, you briefly mentioned this. Maybe we could elaborate a bit more how we kind of got to that number, and if there may be a need for even more if we have multiple doses per patient, is there a possibility that more cells would be needed, is the question.
Right, that's a good question. And I think the short answer is that our processes and products can be used in a scalable and a tunable manufacturing process. So our team is happy to work with you to identify the total numbers of cells that you're targeting. Mm-hmm. And we're happy to work with you to recommend the products and processes, be it a 2D system or a 3D system, to help you reach your target cell needs. And just reading in the literature and the news today, we are seeing that the initial trials are using single dosages to establish some safety, but it looks like follow-on studies showing safety, efficacy, and potentially dose escalation will be required. Absolutely. Thanks, Tim, for sharing that insight. So another question was also regarding our bioreactor. It was noticed that it has a fairly high yield compared to the 2D process. So the question is, why is the yield so high? Right, good question. When cells are cultured on microcarriers in 3D, the surface area to volume ratio is significantly higher than that in 2D flask-based systems like in cell stacks or T-flasks. So when the cells are in the bioreactor and in suspension, you get better fluid exchange due to the continuous mixing, and your media productivity is a lot higher because you're generating more cells per given volume of media when compared to the 2D flask systems. So you're really driving your productivity there. Right, definitely agreed. So I have one more question regarding I guess this is for the bone marrow working cell bank. What is the passage number
at the working cell bank stage?
Right, good question. So the vial cells that customers will receive is what we call our working cell bank, and these are at passage level 2. But I will note that we tend to speak in terms of population doubling level,
Right. Yep.
which really gives a more accurate count for how many times the cells have doubled since isolation. And at the working cell bank level, or passage level 2, the population doubling level is somewhere between 6 and 10, depending on the donor.
Great, thank you, Tim. So another question is, RoosterCollect EVCC, is this a suspension cell culture base media?
I'll try and answer that question. So the idea is that it's an MSC-EV collection media. So the system is designed to use RoosterBio's cells, expansion media, and then collect the EVs using EV Collect. So the basic workflow is seeding your cells into culture in a seed train for 4 days. At that point, when the cells are somewhere between 70 and 80% confluent, you remove the expansion media that is on the cells, the MSCs that are adhered to the 2D flask or the 3D microcarrier system. And then you add in EV Collect, and you will let the cells culture for 2 to 3 more days prior to harvesting that conditioned media for downstream processing.
Right, right. So there was also a question about whether our cells have already been used for an IND application for COVID. I think I'll just say that we are working with various groups on this, and we can't reveal any names at this time, but definitely actively working with groups towards this goal. So, so that, and let's see.
And while you're looking at the next question, Maya,
Yep.
One note to add is that if you are looking to file an IND and want to leverage our products, RoosterBio would be happy to provide the letter of support referencing our master files that you can submit with your IND application.
Yep, absolutely. Thank you, Tim. Another question was regarding our umbilical cord MSCs. What is the passage number for the umbilical cord MSCs?
Right, good question. Umbilical cord MSCs in the literature are found to have a little bit more of a lifespan. In other words, their critical quality attributes are maintained at later passages and higher population doubling level. So the working cell bank of our umbilical cord MSC product will come to you at passage level 3, or population doubling level somewhere between 12 and 14, depending on the donor. And like with the bone marrow MSCs, our cord MSCs are recommended for use for 2 passages after the working cell bank, at which point we perform all of our critical quality attribute testings.
Right, thank you, Tim. So there was a question regarding, again, more questions around passage number. And, you know, when we're transitioning from a 2D culture to a 3D, how do we define a passage number? So Tim, maybe we could give them some insights on how we do this?
Right. In the simplest terms, a passage is considered moving cells from one home to another, right? So one flask to another flask, or from one flask into your bioreactor system, right?
Mm-hmm. Okay, and then another question is the bioreactor. Is the bioreactor the wheel system?
Right, great question. At RoosterBio, we call ourselves bioreactor agnostic. We think that our products and high-level bioprocess recommendations can be adapted to any bioreactor system out there. During our bioreactor development program, we did bring in and evaluate a few different commercially available bioreactor systems out there. For example, the Paul system, Eppendorf, and the PBS Biotech system. Mm-hmm. And for scalable manufacturing and getting really great MSCs after the harvest, we elected to go forward with the PBS Biotech system. It's a scalable bioreactor system. And as I think the person who asked the question, it's that vertical wheel bioreactor system. And we've been routinely executing bioreactor runs at the 15-liter and 80-liter scale. And not only internally, but we've helped to tech transfer 15-liter and 50-liter bioreactor processes to our customers for R&D purposes and already clinical manufacturing efforts.
Yep, great. Thank you, Tim. Yeah, so this last question is similar to what you kind of touched on earlier, which is, have we done comparison with other platforms? So the answer is yes. As Tim mentioned, we have done some studies with the Paul and Eppendorf systems as well. And let me just check to make sure there's no more questions. Let me see. Oh, there's more questions. 1 second. So another question is, if you're using the microcarriers, how would you detach and harvest the final products?
Right, great question. And at RoosterBio, during our development efforts, we came up and prepared high-level bioprocess recommendations for this. At a high level, the harvest will actually happen inside the bioreactor. You start by simply letting all of the cells and microcarrier aggregates settle down. You drain and remove the expansion media. And after doing a quick wash with some PBS, you will then add in the warmed harvest enzyme. We prefer triple select. This enzyme will sit with the cell microcarrier aggregates inside the bioreactor for about 15 minutes with some gentle agitation. And this will actually detach the cells from the microcarriers. That's the first part of the harvest. The second part of the harvest is actually removing the bulk cell solution from your microcarriers, right? Because you don't want your microcarriers or fragments to be in your final cell solution. After coming out of the bioreactor, the cell microcarrier solution will be passed through a 100 micron screen filter. And the idea is that the microcarriers will get caught up in that filter while allowing all of your cells to pass through the filter. And we've demonstrated, very high yields through this filter, and it's a great addition for our unit operation in the harvest process.
Mm-hmm. Fantastic. Okay, I think we'll take 2 more questions since we're coming up to 12:45 pretty soon. So one question is the EV boost. Maybe the question is, can we elaborate a little bit more on how the expansion media, the collection media, and the EV boost is used in a 3D culture? So maybe Tim, you can kind of walk them through a little bit on that again.
Sure. Yeah. So the Rooster Nourish, or the expansion media, is really designed to expand your cells. It's a bioprocess media that's chock full of growth factors and goodies to drive expansion of your cells. And this works in 2D and 3D bioreactor platforms, which is unique because some medias out on the market will work in 2D applications, but need to be adapted for a 3D environment. EV Collect is designed to be a xeno-free, protein-free, and chemically defined collection media. And that's important because most expansion medias out on the market today contain contaminating materials via HPL or fetal bovine serum. Both of those materials inherently have exosomes, which will end up contaminating your final product, which is you're looking to get MSC exosomes or extracellular vesicles. So by switching out to EV Collect for a few days, you know that the stem cells are spitting out exosomes, and your final harvest will strictly be exosomes and extracellular vesicles and particles from your MSCs. The typical collection period that's recommended when using EV Collect is between 2 and 3 days. However, one product that we launched, it's called EV Boost, and it's a reagent that is designed to tune the extracellular vesicle manufacturing process. And by that, I mean it's able to be added at the same time when you switch to EV Collect. And the EV Boost will allow you to shorten the production time.
Yep.
So the amount of particles that you'd be able to collect in 2 or 3 days with EV Collect alone, you can achieve this with EV Boost in 1 8-hour process. Mm-hmm. So really shortening your manufacturing times. I will note that if cells are your product, or you're wanting to keep those cells after EV collection, EV Collect alone might be the way to go. If cells are not your product and you're really just looking to drive EV production, EV Boost is a product that you should certainly consider to boost your EV productivities and shorten your manufacturing times.
Great. Thank you so much for that, Tim. So last question, we still have more coming, but last question, sorry guys. So from one vial of the working cell bank, which for a GMP cell, it's 20 million cells, the question is, how many doses can we produce from that one vial? I think it might be good to just talk through in a 2D process, what is that number, and potentially even in a 3D, Tim, potentially how many cells can we produce in total?
Right. So in a typical manufacturing run, starting from one working cell bank vial from the GMP bone marrow mesenchymal stem cells, it's a 20 million cell vial. And that vial is meant to be inoculated into a one cell stack, so 10-layer flask, and cultured for 4 days, after which these cells are harvested and split into 20 cell stacks for another 4 to 5 days. And in a total of 8 to 10 days time, you will be achieving yields between 3 and 5 billion MSCs. And I will also note that in this 2D process, there are no media exchanges required. So it's a very streamlined, robust, highly productive process that you're going to be using. So depending on your dose size that you're targeting and how many cells you're actually needing, we can actually tailor this 2D manufacturing process to fit your needs. With respect to 3D, the process starts the same. So starting from the 20 million working cell bank, you will do your seed train for the first 4 days in cell stacks. After those first 4 days, you will harvest those cells and inoculate your bioreactor. The bioreactor process will go for 4 days. On day 3, you will add a bioreactor feed called Rooster Replenish, which is designed to replenish any of the nutrients that were consumed or degraded over the first few days of culture and get you to the end of your bioprocess run. After 4 total days or 4 to 5 days in that bioreactor system, we're seeing yields from the bone marrow MSCs north of 30 billion cells per run. So depending on your cell needs and your target requirements for your dosages, we can certainly walk you through our bioprocess recommendations to fit something to your manufacturing needs.
Awesome, fantastic. Thank you so much, Tim. I think that's all the time we have. There's more questions coming, but as we mentioned, we will definitely try to answer all of your questions via email and respond to you directly. And if there's anything else that we can do, feel free to reach out to any one of us on the team. And you can reply to marketing@RoosterBio, or if you have Tim and my email, feel free to reach out to us at any time. And thank you so much, everyone, for attending. We really enjoyed hosting this session. And please give us some feedback if you have any suggestions on how we can improve this webinar. Thank you so much.
Thank you, everyone.
Bye.